All right, this is the third night of the cardiovascular lecture that I'm going to give you. So let's get going. Let's talk about factors that maintain blood pressure. Blood pressure is maintained by a number of things, to include diet and lifestyle changes. But the major factors going on inside the body at any given point in time are as follows. Number one is cardiac output. That is determined by heart rate times stroke volume. Normal heart rate in the adult is 80 beats a minute. Normal stroke volume is the amount of blood ejected from the left ventricle in 1 beat, and that normal amount is 70 cc's. So heart rate 80 times 70 cc's is 5,600 milliliters, or 5.6 liters of blood that is circulated thereabouts every single minute, and that is considered normal cardiac output. That cardiac output falls, what happens to blood pressure? It's going to go down. If cardiac output increases, it's going to go way up as far as blood pressure goes. Number two factor is vessel elasticity. If the vessels are not elastic and cannot stretch and then bounce back or recoil, then blood pressure is going to change as a result of that. Think about patients with atherosclerosis and a bunch of plaque buildup inside their artery walls. Those vessels are not elastic at all. There's great resistance to blood flow, and their blood pressure goes up as a result of that. It's very tough to lower the blood pressure in those patients without taking their blood volumes down to a minimum amount, because we can't do much to the blood vessels. They're inelastic, and if we take the heart rate down, we could throw them into congestive heart failure, because the diastolic pressure is always going to be high. So it's kind of a catch-22 difficult situation. Number three factor is blood viscosity. We've talked a bunch about that. The thicker the blood, the more the pressure is going to go up. Number four factor is hormones. There's many hormones in the body that actually affect blood pressure, to include the thyroid hormones, epinephrine from the adrenal glands, antidiuretic hormone from the pituitary gland, and aldosterone and other steroid hormones that come from the adrenal glands. All affect blood pressure. Fifth factor is peripheral vascular resistance. Remember, the arterioles are the main regulators of blood pressure ultimately, and they also offer the greatest resistance to blood flow by their ability to constrict or to dilate. So peripheral vascular resistance is the amount of peripheral vasoconstriction or resistance to blood flow forward that we are offering by action of the arterioles. Next, we'll talk about some review terms that we have covered in the very first lecture I did to you guys, but they were listed on my objectives to cover with you in the cardiovascular section, so I will review them real quick. The first term is osmosis. Remember, osmosis utilizes no ATP energy, just kinetic energy, which is the universal movement of ions under a normal circumstance. And osmosis moves water or liquids towards a concentrated environment, towards a high concentration of glucose, towards a high concentration of sodium, or whatever concentrated solute is located, whatever concentrated solute is present. Water will move to that region to try to lower or dilute that solution to equalize the concentrations on both sides of a semi-permeable membrane. In this case, we're talking about the capillary wall membrane. Number two, diffusion. Diffusion is similar to osmosis. No ATP energy required, just kinetic energy movement of ions. And in this case, it is not the movement of water, but rather the movement of ions towards water to try to dilute or equalize the concentrations in that solution on both sides of a membrane. So when sodium is more concentrated in the interstitium versus the blood vessel, sodium will move at the capillary level into the capillaries until the concentrations in the interstitial areas and the blood capillaries are about equal. And then movement continues, but in an equal rate. Okay? So remember that diffusion is down a concentration gradient, movement of ions from an area of high concentration to an area of low concentration. Facilitated diffusion is just diffusion using a carrier molecule, and the best example I can give of that is insulin's attachment to glucose to facilitate its transport into body cells. Remember, glucose is a very large molecule and cannot diffuse across the cell membrane because it's too big. So channels have to open up, and it has to actually be transported inside the cell, attached to a carrier protein to get inside the cell cytoplasm. And that is facilitated diffusion. It's still movement of glucose from an area of high concentration to an area of low. It's just attached to a carrier molecule. No ATP energy is required. Active transport, on the other hand, is a movement of ions or solutes up a concentration gradient from an area of low concentration of solute to an area of high concentration. So the best example of this in the body is the sodium-potassium pump. And the sodium-potassium pump pumps sodium out of the cell in a 3 to 2 ratio, and potassium is pumped inside the cell to obtain an unequal concentration across that cell membrane so that when the cell is stimulated by an electrical impulse, sodium channels can open in the cell membrane, sodium rushes in and carries with it the electrical impulse to cause the cell to function as it was made to do. That requires ATP energy, however, to accomplish that active transport process. Next, regulation of blood pressure by the heart and the kidneys. Well, regulation of the heart, or regulation of blood pressure by the heart, is something that you've already learned. Beta-1 cell receptors on the surface of cardiac muscle cells in the heart, if activated, will stimulate the heart to increase their rate, speed of conduction, and force of contraction. So not only are those heart cells beating faster or contracting faster, they're contracting at a faster rate and contracting with more force as a unit to eject more blood. And that is all regulated by beta-1 receptor activation. Now, there are certain drugs that stimulate one or more of those properties more than the others. For example, dobutamine is a beta-1 receptor stimulator, but it's specific in that it only causes those cardiac muscle cells, when stimulated, to actually increase their force of contraction, less so increase their rate of firing, and less so the speed of conduction. So dobutamine is considered a positive inotropic drug. So chronotropic refers to heart rate, inotropic refers to force of contraction, and dromotropic refers to speed of conduction. You need to know those three terms very well because drugs are going to be classified as inotropic agents, dromotropic agents, chronotropic agents, or all three. So make sure that you familiarize yourself with those three terms. The kidneys are able to regulate blood pressure very well as well. The kidneys can sense low blood pressure as blood is carried to them for filtration and will immediately release renin, an enzyme, R-E-N-I-N, renin, into the bloodstream. This renin causes a reaction at the liver to create angiotensin 1. Angiotensin 1 is a very powerful vasoconstrictor, number one, and that's going to raise blood pressure in and of itself. but it continues to travel in the bloodstream and undergoes another chemical reaction at the level of the lungs or pulmonary circulation and becomes angiotensin II. And is, again, a very powerful vasoconstrictor as it travels on through the body, arrives at the kidney nephrons, and causes those kidney nephrons to change their permeability to save water and not eliminate it in urine. So water is retained in the bloodstream. What does that do to circulating blood volume? It increases it. What does that do to blood pressure? It increases it. So that's how the kidneys regulate blood pressure. The kidneys also have the unique ability to change their permeability at any given point in time to water as well as sodium. And remember, where sodium goes, water follows. So if those kidney tubules change their permeability and make sodium re-enter the bloodstream before that blood leaves the kidneys, then water is going to enter the bloodstream in increased amounts as well. And that will increase your blood pressure also. Now, we can give drugs that act on those kidney tubules as well and change their permeability to cause increased elimination of water and sodium. One drug that is famous for doing that is furosemide or Lasix. So we have drugs that can affect the kidney tubules and override that response as well. Other things in the body that can regulate blood pressure include the medulla in the brainstem, along with the pons. The respiratory, the vasomotor, and the cardiovascular system monitors are all in the medulla. And that medulla has the origination of not only sympathetic nervous system fibers, but also parasympathetic nervous system fibers. Remember, I told you that the vagus nerve is cranial nerve number 10, and it provides 80% of the parasympathetic nervous system response if activated. And so it originates in the medulla. And so if the medulla says, whoa, we need to go into feed and breed system override instead of fight or flight, then the parasympathetic nervous system sends its impulses along cranial nerve number 10 to the heart to decrease heart rate, force of contraction, and speed of conduction. It goes to the blood vessels, tells them to relax or dilate. It goes to a lot of different body areas to cause just the opposite of what we think of as a sympathetic nervous system response. Well, if blood pressure is too low, just the opposite occurs. The sympathetic nervous system fibers leaving the medulla will be activated to go to the lungs, the heart, and the peripheral vessels. Speaking of the peripheral vessels, we've learned over and over again that alpha-1 receptors lie in the smooth muscle of peripheral blood vessels, in that muscle layer, the middle layer of blood vessels. It's also alpha-1 receptors are also located on the membranes of the precapillary sphincters. Well, we have another set of sympathetic nervous system receptors there as well that I haven't talked about until right now, and they are the beta-2 receptors. Previously, I've learned that beta-2 receptors are located in the lungs, in the bronchial smooth muscle. And if activated, those bronchioles will dilate, won't they? Well, the same thing happens when beta-2 receptors in the peripheral arterioles and precapillary sphincters are activated. They will dilate, and that lowers blood pressure or increases blood flow to that area, okay? So for example, if you go into fight or flight mode and the sympathetic nervous system is activated, what blood vessel areas would you want to dilate and shunt blood to? Well, your brain, heart, and lungs to preserve life, as well as the skeletal muscles so that you have increased blood flow to your skeletal muscles to outrun the lion or outrun the bear, as in the primitive times. When you went into fight or flight mode, something was chasing you, something was after you. And so you needed increased blood flow to your skeletal muscles. So that's why we have those beta-2 receptors lying in those muscle areas. Now let's shift gears and go to the part of the lecture where we're going to talk about the electrical conduction system of the heart. All right, so what I want you to do is get a good picture in front of you, either in your textbooks or on Wikipedia or somewhere on the internet that shows a good picture of the electrical system of the heart. Once you have that, let's move forward with the SA node. The SA node is in the upper portion of the right atrium. It is the pacemaker of the heart. That is where all of the stimuli under normal circumstances are generated for that entire heart muscle. The internodal conduction tracts then leave the SA node. There's 3 of them through the right atrium, and 1 of them, Bachman's bundle, that goes to the left atrium and branches out and spreads that impulse throughout both atrial networks. Atrial networks are linked together so that the atria, under normal circumstances, contract together. So if stimulated, the entire left and the entire right atrium contract at the same time. That is called a syncytium. Syncytium. A syncytium is a network of fibers, and the atrium, both left and right, is a syncytium. Next, the impulse is carried down to just beside the mitral valve, and that is called the AV node, atrioventricular node. The SA node, by the way, is called the sinoatrial node. The AV node is an area of very high importance because this is where the impulse is immediately slowed down for a period of time. And that slowing of the conduction through that region gives the atria time to contract and expel as much blood as they can into the ventricles before the ventricles are allowed to contract. Because if that impulse wasn't slowed, it would go zip all the way through the atria and all the way through the ventricles, and the whole heart would just pump as one unit, a one-stage pump. Well, in that case, blood would squirt out both ends of the heart, wouldn't it? And we want blood to go forward. So it's a two-stage pump. The atria contract following stimulation. The impulse is stopped and slowed down briefly at the AV node, gives that ventricle syncytium time to fully distend with blood so that it can contract once it's stimulated and eject an adequate amount of blood. So that's what's going on in the AV node. The next structure that you see is the bundle of His. That bundle of His carries the impulse from the AV node to the interventricular septal wall. That's the purpose of the bundle of His. The impulse immediately speeds up once it hits the bundle of His. Next, locate the left and right bundle branches. The left bundle branch will carry the impulse down the septum to the left ventricle, down the apex first, and then back up, because we, remember, have the semilunar valves are at the top of both ventricles, not at the bottom. So blood is expelled out the top of both ventricles, not out the bottom. Okay? So the ventricles have to contract in the septal wall and the bottom of the ventricles first and shove blood up, squeeze and twist and wring itself out to squirt blood out the top of those two valves, the pulmonic and the aortic valve. Next, locate after the bundle branches the Purkinje fibers. These fibers spread throughout both ventricles and send the impulse very rapidly through both ventricles, so that once both left and right ventricular cardiac muscle fibers are stimulated, they will pump together as one unit or syncytium. The prime function of the electrical conduction system is to transmit electrical impulses to the working cardiac muscle cells. So understand that in the heart you have conduction cells and you have cardiac muscle cells, two very different types of cells that make up the heart. Okay, the impulse travels very fast through the atria. It only takes 3/100ths of a second for the impulse to travel through the left and the right atrium. Now, now that I think about it, I may have messed up and told you that the AV node is by the mitral valve. You might check your notes. The AV node is actually by the tricuspid valve in between the right atrium and the right ventricle. So I hope I didn't misspeak, but I'm just clarifying that right now. The AV node is by the tricuspid valve, not the mitral valve. The AV node, working backwards and taking a further look at it, consists of three regions. You have the smaller upper internodal region, and you have the middle nodal region, which is the large central area of the AV node when you look at it in pictures. And this is the area that is actually involved in slowing down conduction through the AV node. And then you've got the smaller lower nodal region that attaches to the bundle of His, where the impulse speeds back up and is unimpeded again. The SA node, the AV node, and the Purkinje fibers all are potential pacemakers of the heart. The SA node generates impulses at 60 to 100 times a minute. The AV node generates impulses at a rate of 40 to 60 times a minute. And the Purkinje fibers is our last-ditch effort at a heartbeat and stimulates the heart at a rate of 20 to 40 beats per minute, or 20 to 40 impulses per minute. So you can see that the SA node, because it's generating impulses faster, will be the major pacemaker of the heart. But if the SA node fails from a right ventricular infarction or what have you, then the AV node may be the next best option. Well, when you hear of junctional rhythms, this is an impulse that begins at the AV node and will sustain life in most cases, but we lose that atrial kick with AV junctional rhythms because those four tracks that travel through the atrium are no longer stimulated by the SA node to fire. And the impulses will not go backwards. They will only go forwards. So the only thing that is stimulated is from the AV node to the bundle of His, the interventricular septum, and then both ventricles through the Purkinje network. Now, if something happens to the SA node and AV node, and they're both knocked out, then we have to rely on the Purkinje fibers just in the ventricles. And a rate of 20 to 40 beats per minute is not enough to keep most adults conscious. So this is very inadequate rate. And in most cases, you'll be doing CPR on these folks because it will not be enough of a heart contraction to sustain life. The other thing is that when you've just got the Purkinje fibers firing, the only thing that is actually stimulated to contract are the ventricles, not the septum of the ventricles. So the outer, posterior, and inferior walls of the ventricles is it. So cardiac output is not only decreased by how slow the rate of contraction is at 20 to 40 beats per minute, but also very ineffective force of contraction because the interventricular septum is not involved in that contraction any longer. Just throwing some numbers out to you. The AV node slows the impulse from 0.6 to 0.12 seconds is how much it slows it down. You don't need to know those numbers right now, but that's how much the impulse is slowed. The next structure, the bundle of His, speeds that impulse back up to 0.03 seconds, which is exactly what it was through the atria by the SA node. The right and left bundle branches travel down the interventricular septal wall, and because the left branch has to travel over more muscle, because the left ventricle is so much bigger than the right ventricle, it splits off into two branches itself, and it's called the left anterior and the left posterior fascicle. So if you've ever heard of a doctor or a paramedic or a nurse talk about, oh, he or she has a left fascicular block, they're talking about one of those two branches being blocked, and you can see that on an EKG. Either the anterior fascicle is blocked or the posterior fascicle is blocked, and you can see those things when you look at a 12-lead EKG. The Purkinje fibers, just because they are slow as pacemakers doesn't mean that electrical impulses don't travel very, very rapidly through them under normal circumstances. Because the ventricles are so large, an impulse has to kick up speed and go into overdrive really, really fast to get the whole thing stimulated so it can contract on a very efficient basis. That impulse travels through the Purkinje fibers at a rate of 0.01 seconds, or 1/100th of a second is all it takes. That is moving. And so the entire heart conduction from SA node to Purkinje fibers under normal circumstances only takes 0.2 of a second to get through the entire thing. Even when you take into consideration it's slowed down by 0.12 seconds, up to 0.12 seconds at the AV node. Now, that's the normal conduction through the heart, but there have been studies done on hearts that don't conduct those impulses normally, and there have been 3 alternate pathways that have been identified in patients' hearts that are abnormal pathways that have specific names, and you need to know them. So these can actually cause pre-excitation syndrome, is what we call it, or ventricular pre-excitation syndrome. And the most common of these accessory pathways, kind of like anastomoses or collateral circulation, when I talk to you about blood vessels, these are alternate pathways around the AV node. So what happens is the worst. The AV node is no longer involved in that conduction to slow the impulse. So the heart speed of conduction from the atria to the ventricles is altered. And so let's take a look at these. The accessory pathways that go through the AV node are called the bundles of Kent, capital K-E-N-T. And they consist of conductive fibers, just like are found in the SA node and AV node, and they bridge the layer between the atria and the ventricles, which is normally impervious to electrical conduction. So when they are present, they provide a pathway that is going to be preferred for that heart because there is no impedance to electrical conduction through that region versus the AV node. Now, between the posterior wall of the left atrium and that of the left ventricle, there is a pathway called the Wolff-Parkinson-White conduction pathway. Okay, and that one is a very common alternate pathway that we see, and when we teach you EKGs, we will show you what WPW, or Wolff-Parkinson-White syndrome, looks like on the EKG. Between the posterior and atrial ventricle walls, we can also see in the septal region another alternate pathway, and that is actually another type of WPW syndrome, only it's posterior wall WPW conduction pathway syndrome. And finally, the third type of Wolff-Parkinson-White abnormal conduction pathway is called type B. And this occurs when the fibers run from the anterior free wall of the right atrium to that of the right ventricle along the anterior wall. And that's called type B WPW. Now, do you need to know the different types of WPW? No, you do not. Just know that there are 3 different types. They all are alternate pathways around the AV node, which is important for you to know. So the impulse is not slowed at the AV node, okay? When you take into consideration that the SA node is going to produce a bump on our EKG called the P wave, and then there's going to be a slowing of the conduction through the AV node, which is just a flat line on our EKG. And when the impulse picks up again in the bundle of His and down the septal wall, the impulse will produce the QRS complex as the impulse travels over, down to the bottom of the heart, and then back up. That's what produces that wavy QRS complex, okay? And then following that, we have an isoelectric line because the heart's resetting itself, waiting to undergo the next cardiac cycle, right? And while it's resetting itself, the sodium-potassium pump is pumping sodium out, potassium in, and calcium is returning to the interstitial areas into the sarcoplasmic reticulum inside cardiac muscle cells, so that we get a bump after the QRS called the T wave, which is repolarization or the movement of potassium out of the cell. And then we're ready to accept another impulse, starting with the P wave, which is the SA node conduction through the atria, the isoelectric line following that, which is the AV node conduction, slowing down the impulse, and then the QRS complex as the impulse enters the bundle of His, down the septal wall, and up the ventricles through the Purkinje fiber networks, okay? So when you consider that, and we talk about these alternate conduction pathways, that pathway is not going to slow the impulse through the AV node. So what happens to the P wave? It's right on top of the QRS on the EKG. So we get what we call delta waves, which are P waves right on the front half of the QRS. And that's why it is produced, because the impulse is not slowed through the AV node. I hope you all understand that. All right, next, atrio-His fibers can also have alternate conduction pathways, and these are called James fibers, capital James, James fibers. These connect the atria with the lowermost part of the AV node near the origin of the bundle of His. Now remember, when I talked to you originally about the structure of the AV node, it's that middle large portion of the AV node that actually slows the impulse. The bottom part of the AV node that connects to the bundle of His no longer impedes electrical conduction. So the impulse is going to be fast around that pathway as well, the James fibers. And this is called atrio-His pre-excitation, when the fibers travel along James's fibers. Nodoventricular and fascicular ventricular fibers. The nodoventricular and the fasciculoventricular fibers, also known as Mayhem fibers, capital M-A-H-E-I-M. Mayhem fibers, M-A-H-A-I-M fibers, okay? The Mayhem fibers are accessory conduction pathways that provide a bypass channel between the AV node and the ventricles. And they do it in the following locations. First of all, between the lower part of the AV node, once again, and the right ventricle. These are called nodoventricular fibers. of the mayhem fibers. Next, there can be an alternate conduction pathway between the bundle of His and the ventricles, which bypasses the interventricular septum left and right bundle branches. And this AV conduction is called nodoventricular and vesiculoventricular conduction pre-excitation. Do you need to know that? No. But know about mayhem fibers, providing alternate pathways from the AV node to the Purkinje network, and know that James's fibers provide an alternate pathway from the bottom part of the AV node around the bundle of His. Okay? All right, let's review cardiac muscle cells. Get a good picture of cardiac muscle cells in front of you, and we'll talk about those next. The heart is composed of cylindrical cells. They're shaped kind of like a tall can of asparagus, if you will, that particularly divide at their ends into two or more branches. These branches can connect with adjacent cells by way of intercalated discs. Don't forget those. The intercalated discs are the way in which cardiac muscle cells communicate with each other. That makes the entire network of atrial cardiac muscle cells and ventricular muscle cells two separate networks called syncytiums. These membranes contain areas in the areas of the intercalated discs of low resistance to electrical impulses. And these low areas of resistance in the region of the intercalated discs are called gap junctions. They permit very rapid conduction through them so that one cardiac muscle cell is stimulated, they all get stimulated at the same time. If the impulse was slowed at all in this network, the cardiac muscle cells would contract in a wave pattern, not as a unit. So that's very important. The ability of cardiac muscle cells to conduct impulses together is called the property of conductivity. You need to know that. Cardiac cells are enclosed in a semi-permeable cell membrane that allows certain charges, particles, to transcend them, to permeate them, which include, as we've talked about 100 times, sodium, potassium, and calcium primarily. Those are our three ions that cause cardiac muscle cell stimulation and cardiac muscle cell repolarization. And this causes the generation of electrical impulses as well as cardiac muscle cell contraction. There are two basic types of cells in the heart, and I mentioned this just a few minutes ago. The working cardiac muscle cells, which you see pictured in your textbook or on Wikipedia, whatever you're looking at, and the specialized electrical conduction cells that we just went over a few minutes ago, starting at the SA node. The myocardial cells have a thin muscular layer of atrial wall and a much thicker ventricular wall. These cells contain numerous myofilaments that consist of contractile protein filaments in units, and these, remember, are called actin and myosin, the major players of cardiac muscle cell contraction. The myofibrils give those muscle cells the ability to contract very hard, very forcefully. So muscle cells have the ability of conductivity and contractility. The force of contraction increases in response to certain drugs. So force of contraction is an inotropic property, and I've already told you a drug called dobutamine specifically causes the muscle cells to increase their force of contraction. Other drugs can also stimulate the cardiac muscle cells to work in different ways as well. Digitalis is a positive inotropic drug. Sympathomimetics, which are epinephrine and norepinephrine, for example, and physiological conditions can also change these cardiac muscle cells' ability to contract and relax harder or less so. Increased venous return to the heart. If we dump a bunch of excess blood volume back into the right atrium, it's eventually going to get to the left ventricle and cause increased force of contraction, because the more the cardiac muscle cells are stretched and filled with blood, the harder they're going to contract. Okay? That property is called Frank-Starling's law of the heart, and it is simply put, more in, more out. Cardiac muscle cells will contract to their hardest ability when they are adequately stimulated. Now, what will make them contract harder is the greater the distension to a point, the harder they will recoil and contract. Okay? It's just like taking a rubber band. If you take a rubber band and stretch it to its fullest potential and let go of it, it's going to snap a lot harder than if you just barely stretch it and let go of it, correct? But if you overstretch it, what happens? That rubber band breaks. Same goes with the heart. To a point, that heart will contract harder the more it's distended with blood, to a point. But if you over-distend it with blood, there is a breaking point. That heart will no longer be able to contract forcefully. It'll just give up and just sit there and fibrillate, or just halfway contract and just squirt some blood out and sit there full all the time with too much blood in it, which in and of itself causes a lot of problems. Exercise, when we have increased oxygen demands in the body from trauma, exercise, exertion, anything like that. Emotions can increase the workload on the heart. Hypovolemia and anemia, a lack of oxygen being carried through the bloodstream, will cause increased force of contraction. All of those things will change how the heart muscle cells contract and respond. In contrast, there are drugs that do the opposite effect. Beta blocker drugs, such as metoprolol, propranolol, atenolol, all of those drugs oppose the beta-1 receptors being activated and will cause the heart to have a lesser force of contraction, slow their speed of conduction through them, and also decrease their rate of firing, so decrease the heart rate. Potassium, if administered, can actually stop the heart because it opposes the sodium-potassium pump when you administer too much potassium to your patient. Pathological conditions such as infections, pericardial tamponade, shock, hypothyroidism, all of those things definitely affect the ability of the heart to pump effectively and stronger. The specialized cells of the electrical conduction system do not contain contractile units, actin and myosin. The myofibrils are not present in the electrical conduction system cells, the SA node, AV node, and so on. They do, however, contain more gap junctions than the myocardial cells do. So the impulse is spread preferentially through the conduction cells first before the cardiac muscle cells follow with their stimulation and contraction. If you do not remember how cardiac muscle cells contract, definitely go back and look at a video on YouTube and review that section in my very first lecture, because I went over that in detail, how the actin and the myosin myofilaments get together and cause the cells to contract. Remember, troponin and tropomyosin also cause a major role in contraction and relaxation, as well as calcium. So make sure that you go back and review that at this time so that you understand the role of calcium, actin, myosin, troponin, and tropomyosin. In particular, note the role of calcium, because we're going to be giving calcium channel blockers to patients to change that heart's force of contraction and speed of conduction. And you need to understand how it affects cardiac muscle cell contraction if we block the calcium channels.